So don't watch me. I'm going to move around. If you snore, I will come by and sit beside you, okay? So we'll try not to make it too boring tonight. Also, this is a conversation. I don't want to sit here and listen to myself for the next hour or so. So please, if you have questions, raise your hand. We'll try to address those as they come up, okay? Okay, so before we dive into this, I'll just kind of give you my why. Like, why invest the time in learning about epigenetics? And the reason for that is, well, my job is to optimize a patient's health, cure disease, and when that's not possible, try to alleviate symptoms as much as possible, morbidities, allow them to function as well as possible in life. And that's where epigenetics can be a very good tool or vehicle for change. Giving someone data that says, you know, when you sleep 6 or less hours a week, 700 of these genes that have to do with diabetes, heart disease, things like that get switched on. So when someone sees that in a research article, sometimes it really lightens them up and creates change. And that's all about what we're doing, is trying to figure out how to elicit change in people. And a lot of that has to do with lifestyle management. And I can tell you, practicing for 10 years now, and before that, I was a personal trainer, ran some health clubs for another 10 years. Trying to get people to change lifestyle behaviors is very, very difficult. We all tend to get ingrained in kind of the same routine. Convenience usually trumps health. And so if something's more convenient, we're more likely to do that instead of going the extra mile or doing something extra to be healthier. So trying to get people to weigh the balance of inconvenience and health, it's challenging. So epigenetics can be a tool in order to do that. In the last 10 years, I've been researching, using this with patients. It has been a very, very useful tool. I'll give you two big, cases that have been the most aha moments for me. Actually, one of my first cases where we looked into genetics was a 30-year-old woman who had had 10 miscarriages. And if you've ever had a miscarriage, you know how hard that could be. And to have 10 of them was really traumatic. But we started looking into some of the genetics of this, and she had this particular variation of her MTHFR gene, and it required really high doses of particular nutrients, methylfolate and methyl B12. And she eventually was able to actually get pregnant, maintain the pregnancy, and had a healthy baby boy. So that was one instance that really kind of took my attention, like, hey, this can be a real game changer. And this was 8 years ago, and we've learned so much more just in that time period. The next case was actually a local woman. She was a fitness instructor and ate like a bird. She would teach 5 to 7 classes, cardio classes per week. Barely ate enough to let anyone survive, and still could not lose weight. And she had about 100 pounds to lose, if you can imagine that. And everything she tried just didn't work. So we did some genetic testing and found, her body elicits a very high stress response. And anything that's going to stress her body is going to shut it down. It's going to flip the switches to conserve energy. And that's what she was doing by doing all this cardio. So when I told her, you actually need to do less cardio, exercise less, and eat more, well, that just didn't make any sense, right? Because calories in versus calories out, that algorithm just doesn't make sense if you're trying to flip it like that. But she did. I said, give it 6 months. Let's just try this and see what happens. Sure enough, she lost 60 pounds in 6 months. And last I checked, it's been a couple of years, but she's down almost 100 pounds. By exercising less, weight training more, less cardio, and eating a good balanced diet, getting enough calories in so her body's not stressed. So a couple of cases there for you. Let's dive into this. So how does genetics work? So if we're going to talk about epigenetics, we need to kind of have a basic idea of how genetics work. So typically, you've got your DNA. We're going to call that your hardware if we're using a computer analogy here. That hardware is going to get transcribed into messenger RNA and then translated into a protein. Most of the time, that protein is some type of enzyme. An enzyme is a catalyst for a reaction in your body. Okay? Those reactions, thousands of them are going on per day, and that's what drives processes in the body. The Human Genome Project, you've probably heard of it. It was completed in 2003. They had their first rough draft in 2000. How many genes do we have? Roughly about 24,000, which was a big surprise. Most scientists were thinking, oh, well, we're going to be over 100,000 genes there, but it was much less. How many base pairs? So a base pair is a adenine, thymine together, or cytosine, guanine together. That's considered a base pair. And we do have 3 billion base pairs, but only about 3% of those base pairs are what we call genes that spell out something, that actually produce like an enzyme or a protein that's going to be part of a process. And these instructions only differ about 0.1% from one person to the next. So that's 1 per 1,000 genes. So we used to think the other 97% of those genes were just junk genes, okay? Like they didn't do anything. Well, that was wrong. That's actually where most of those epigenetic changes are lying, and that's what we're finding out now. So what's epigenetics now? It's the study of changes in gene activity that don't involve changes in your DNA. So if your genetics, your DNA is your hardware, epigenetics would be your software. So it's telling your hardware what to do. And epigenetics can change how your DNA is expressed. And this is the big part. And we'll talk about like identical twins. And if you watch identical twins as they get older, especially if they live in different places, how they will eventually start to look a lot different and have different diseases. And that's the epigenetics. And you're changing your genetic expression daily, perhaps even hourly, from the food you eat, the stress level, air you breathe, how much you sit or lack of movement, exposure to toxins, and sleep. So everything in your environment is affecting how your genes are going to be expressed. Some of the ways that we know that genes can be altered, their expression can be altered, are DNA methylation, histone modification, chromatin remodeling, and non-coding RNA. Now, for this lecture, we're going to focus mostly on DNA methylation because, honestly, it's been around the longest. It's been about the mid-'80s when this started being researched heavily, and we just know the most about it right now. Okay, so this is, as far as methylation goes, it's pretty simple. When you take a methyl group, which is just a carbon and 3 hydrogens, and you add it to a base pair, typically a cytosine, you silence that gene. So that gene can actually not express the protein that it's meant to express. Now, if that's a tumor, if it's an oncogene, a cancer-causing gene, then that's a good thing, right? We want to silence that. We all have we call them good or bad genes, but we want to silence as many of those sort of bad genes as we can. This also goes into a little bit of the histone modification. Histone is just a conglomerate of those genes together. It's kind of a way of increasing the amount of, or decreasing the amount of space. And then you can have these little tags you'll see up in this right-hand corner. These epigenetic tags also affect the way that the histones get expressed. Okay, so methylation, I call this our mechanic. So the methylation cycle, which we'll see in the next slide, is a way to produce that methyl group that attaches to that base pair to silence that gene. So we're going to focus on how do we produce more methyl groups, and that's through a process called methylation. So methylation is going to be like our mechanic. If you add a bad gene, we add a methyl group to that, you silence that gene.
Can I ask a question before
Sure.
you on? So if you have a methyl group and it goes to a base pair that's the beginning of a good gene, is it going to silence that?
It can, yeah. It's going to silence any gene that it's attached to. And we still don't know what causes that methyl group to attach to that specific base pair.
But methylation is a huge problem among our population.
Oh, absolutely, because
HFR is a huge problem.
there's so many things that affect methylation and that this process here. So this is actually a simplified version of the methylation cycle, if you would believe that. But everything in color there is an enzyme. That enzyme, remember, is made by a gene. It's produced by a gene, right? So what we do here, this is kind of where I start with the genetics, is we'll run genetics on this to see if someone has a variation in the gene that makes these enzymes, okay? Everybody follow me so far? If you have a variation in the ability to make that particular enzyme, it's likely you're going to make less of that enzyme. So let's talk about MTHFR. This is the one here in blue right in the middle. So this is what we call the rate limiting step in this process. It's the most important step. So if there's a problem with this particular enzyme, you're going to have big issues, okay? And we know because this is the rate limiting step, if you look in PubMed, there's over 5,000 articles just on this one particular enzyme and gene, okay? So it's really important. And we know from all that research that if you're homozygous, so if you have two variations out of two, maybe you get one from your mom, one from your dad, if both of those are, we don't want to call them mutations because they're not really mutations in that sense, but we'll call them variations, something abnormal, okay? If you have two of those variations, then you make about 70% less of that enzyme than someone who doesn't have those variations. So you can imagine if you're making less of that enzyme, that's the most important step in this process, then you're less likely to, the end result is making those methyl groups, which is over in this right-hand side.
So you just have one copy.
Well, it can be a little complicated because there's multiple MTHFR genes, and the two particular ones, if you're compound heterozygous, so yeah, then that can be somewhere about 40 to 50% of reduction in the enzyme. Yeah. And then if you look here too, if you've got variations in that enzyme and other ones in this process, then that effect can be compounded. We still don't know really what, if you have one of these and one of these and one of these and what that results in, because there are a ton more variables and there's a lot we still don't know. We're just at the tip of the iceberg here. We don't know really how big this iceberg is yet. Okay, so we start with identical twins. They look, identical, right? Very cute. But as they get older, they start to change a little bit. Look a little different. What's going to do that? So let's go through some examples, stress and epigenetics. So this, unfortunately, looked at suicide victims with a childhood history of abuse. And what they found is if that person had a history of abuse, they had a higher risk of these changes that led to diminished capacity to effectively respond to stress. They had elevated levels of anxiety in adulthood and suicide. The next study showed that children of women who were abused during pregnancy were significantly more likely to have children with changes that altered their response to stress. So now we're looking at multi-generational changes here. Not just the children of the women, but the children of the children had these changes passed on. So now we're seeing these epigenetic changes can actually get passed down, just like your DNA, to your offspring and so on. The Dutch is a good example of this in the famine. They take great records, and they've been able to see those changes, epigenetic changes, being passed on from generation to generation. And we'll talk about that in a little bit here. The mind and epigenetics. This is really fascinating because this might actually explain the placebo effect. Okay, Dawson Church, with a great book, The Genie in Your Genes, found that heart surgery patients who had a strong social support network, a spiritual practice, had a mortality rate 1/7 of those who didn't. And studies showing that meditating for just 30 minutes per day can boost your mood as much as many common antidepressants. Older people with positive attitudes have a 23% lower risk of death from cardiovascular disease and 55% than those with negative attitudes. This is a big one here. We know that the one thing that will allow you to live longer is your attitude. Optimistic people tend on average to live about 11 years longer than pessimistic people. That's the power of your mind right there. And we think that this change in mindset's actually creating a change in genetic expression, which may explain the placebo effect. The placebo effect can account for about a 20% change in studies. And we really haven't figured out why that is. This may be able to explain it. If you think you're getting the medication that you need to benefit you, then you're changing, that mindset is changing your genetic expression. And the end result can be positive. Okay, diet and epigenetics. This is a great way to change your genetic expression. Folic acid, this is a big one as part of that methylation cycle. You need folate to run that. You can get folate from your diet. Green leafy vegetables, uncooked green leafy vegetables are a great way to get folate. Folate is not the same thing as folic acid. I just want to make that clear because you'll see those used synonymously and they're not the same. Folic acid is a synthetic product that is used to refortify grains and other things that have been refined. That refining process takes the B vitamins out, and so we have to put those B vitamins back. So we typically use a very cheap synthetic form of that folate. Well, we know about 30% of the population has a variation in the ability to take folic acid and actually convert it into folate. And those are conservative numbers. Some estimate it could be as high as 50%. So diet and epigenetics. So we all have genes called agouti genes. And so they use agouti mice as a way to look at some of these genetic variations. When a mouse's agouti gene is completely unmethylated, so remember, we're taking that methyl group away, that carbon and 3 hydrogens, they end up being yellow and fat or obese. And they're prone to diabetes and cancer. When you methylate that, so you add that methyl group onto that particular gene, their coat is brown and they're skinny and they have a low risk of cardiovascular disease. So pretty easy to see, fat yellow mice. Okay, they've got the unmethylated form. Skinny brown mice, they've got the methylated form. But we know a fat yellow mice, if they're genetically identical, it's just one's methylated and one's not. But if you feed that fat yellow mouse a high methylated diet, so lots of methyl donors, so those uncooked green leafy vegetables with lots of folate in them, then they'll have babies that are brown and skinny. So even though they're, genetically they're unmethylated, they can still have offspring that benefit from the methylation. And you can see here too, some of these methyl donors include B12 and folate, but choline, methionine, and zinc are also nutrients that are part of that cycle as well. Okay, more on diet and epigenetics. This is from the Dutch famine, 1944 to 45. Showed increased rates of coronary heart disease and obesity after maternal exposure to the famine during early pregnancy compared to those who didn't. So we know now that less DNA methylation in this particular gene, this insulin growth factor, which is how we actually measure growth hormone, results in this increased risk of coronary heart disease and obesity. Another study published in 1986 showed that if pregnant women ate poorly, so high fast food, low fruits and vegetable diet, the child would have a significantly higher than average risk for cardiovascular disease as an adult. So again, we don't see a lot of this play out until these children are adults. In 2016, Johns Hopkins found that a high-fat diet, low-carb diet could actually open up chromatin and improve mental ability in some of these inhibitors. So I know it's kind of a fad right now, a ketogenic diet, but in some circumstances, this can actually help mental focus and ability by affecting your epigenetic expression. And then other studies have shown that certain compounds within foods could protect against cancer by adjusting the methyl marks. That again goes back to the selenium, the zinc, the folate, and the B12. Toxins and epigenetics. So chemicals like BPA. This was popularized a few years ago. BPA was a bad thing. It's found in a lot of plastic water bottles. It has an estrogen-like reaction in the body, so it creates a lot of issues. But the BPA in the body will actually reduce methylation. So again, we've got these agouti mice, and when mothers were fed a high BPA diet, their babies were more likely to be yellow and obese, again, which means that they were unmethylated. So this BPA was affecting the ability to make methyl donors, that's carbon and 3 hydrogens, or the ability of it to attach to the base pair. However, when the mothers were fed BPA along with a methyl-rich diet, they had babies that were brown and skinny. So even though the toxin was there, if they could overcome that toxin with the proper diet changes. And that has been shown true in humans as well. And that's something too, like, it's hard because we live in an increasingly toxic world. We import billions of pounds of synthetic chemicals from other countries every year. There have been studies that looked at the cord blood of newborn babies, and they've got on average 200 to 300 different synthetic chemicals in their cord blood. So babies are being exposed to chemicals in utero. We're going lifetimes of exposures now to high levels of plastic and phthalates and BPAs and all these endocrine disrupting hormones or chemicals. And there's a lot we can do about it. We can clean up our diet, the air that we breathe, the water that we drink, all those things. But to some extent, there's only so much we can do individually. We find BPA on the mountaintops of every mountain in the world. So it's in our atmosphere. It's ubiquitous in our environment. But we can overcome some of that by changing our diet, by adding methyl-rich foods into our diet. Okay, researchers, University of Sweden in 2013 showed significant changes in those who smoke compared to smokeless tobacco. So it was actually the thing in the cigarette. And that created hypomethylation in gene regions over 20 gene regions linked to heart disease, cancer, hypertension, and cataracts. Another study published in 2013 showed that mothers who smoked had children with several regions of this hypomethylation, and they had higher rates of ADHD, major depressive disorder, and substance abuse during adolescence and childhood. So again, mothers, and this was before pregnancy, not while they were pregnant. So mothers who smoked prior to getting pregnant also affected their children later on. Air pollution could also affect these methyl tags on DNA and increase risk of neurodegenerative diseases.
Okay, I'm not leaving this building.
Yeah, right? Yeah. We have masks in the back for everyone. No, I wish, but yeah. Okay, sleep and epigenetics. This is one that really fascinates me because most people, you ask them how they sleep, they're like, okay. And we just kind of brush it off like, not getting enough sleep, or even trying to function on very little sleep is almost a badge of honor. Like, I only need 4 hours of sleep. Well, that's not true. A study conducted in the University of England, this was 2013, showed that only 6 hours of sleep per night for 1 week, and then they were kept up for 40 consecutive hours, resulted in an average of a change in over 711 gene expressions. Most of this had to do with heart disease, diabetes, and cancer. So that's just 1 week. Good news is they took those same set of people, they allowed them to sleep for 10 hours per night for 1 week, kept them up for another 40 consecutive hours after that, and saw that most of those genes, about 60% of those genes, were switched back from where they were originally. According to the CDC, 30% of civilian adults in the U.S., that's over 40 million workers, report getting 6 or fewer hours of sleep per night most nights of the week. That's pretty significant. Exercise and epigenetics. This is a really fascinating study. I want to meet the scientists who came up with this idea. So in 2014, what they took are cyclists and only using one leg for 45 minutes, 4 times per week for 3 months. They took muscle biopsies of both legs, but they had them only use one leg, okay? And they measured the methylation status in those biopsies in both sets. So same person genetically, right? Even same food, same other environmental exposures, just one side exercised and the other side didn't. And what they found was over 5,000 sites on their genome showed new methylation patterns. That's a good thing compared to the non-exercise leg. Most of these genes looked at a role in some type of energy metabolism, insulin response, and inflammation within muscles. Unfortunately, they didn't look at how long those changes persisted after exercise. I'm sure because the individuals didn't want any more holes punched in their muscles. But fascinating studies, great idea. We see this in nature. So royal jelly, which is a complex protein, and it's got a lot of B vitamins, especially B12. So genetically, the worker bees and queens are genetically identical. But queens get the exposure of this royal jelly, and they become queens instead of worker bees. Medications and epigenetics. So this is a pretty new field right now. There's not a lot of medications out there. This is one that's been approved for myelodysplastic syndrome. They did show some progress. 16% of people who received it responded either completely or partially. Myelodysplastic syndrome is it's kind of a mild form of bone marrow suppression. You see low levels of platelets, white blood cells, red blood cells. 2/3 of the patients did not require blood transfusions after the study. So it's potentially looked at as a chemotherapy agent as well. So what does the future hold about this? So you can see these are the publications over the years, since 2000 when the first report of the human genome was sent out. These are the amount of publications done on epigenetics per year. You can see from about 2012-13 to 2018, we've pretty much done as many in that short amount of time as the 10 years prior to that. So this is just exploding right now. Tons of publications are coming out every month. Yeah.
What's the associated cost with measuring epigenetic changes? Like, is this something that costs have gone down for over the years, or could we see that in the future for more studies or even access for a common person to get?
Right. Yeah, so right now the step is, genetic testing is getting so cheap that, for $100 in 23andMe, you can get, 600,000 base pairs looked at, okay? So that kind of testing would have cost you thousands upon thousands of dollars just 10 years ago, okay? And so that was kind of the first step. Second step is like being able to see these epigenetic changes. And that testing is pretty expensive, and it's mostly done at universities now. But it's getting cheaper and cheaper, and as more and more studies come out and more companies are jumping in, dumping lots of money into it, then that's going to become more accessible to consumers, either directly to consumers, or I think in the next 5 or 10 years, you'll be able to walk into your doctor's office and get your whole genome mapped out and be able to see genetically and epigenetically as well, like what medication might be best for you. If you're trying to make a decision between which classification of antidepressant you want to try, we can already do that now. We can already kind of start to narrow some of that down to which ones you're more likely to respond to, which ones you're more likely to have reactions to or adverse side effects to. So I think that's there. We're right there on the edge of being able to make that accessible to consumers. Yeah, good question. Okay, how do you optimize your epigenetics? Well, it just comes down to basic lifestyle modification, right? So eat clean food, more whole foods, less packaged foods. If it can sit on the shelf for 2 years, it's probably not the healthiest thing for you. Organic meats and dairy, lots of fruits and vegetables. Again, that's where we go back to those methyl donors, lots of uncooked green leafy vegetables. Dirty Dozen, Clean 15. This is a great list by the Environmental Working Group. This is, if you can't afford, especially here in Fairbanks, trying to buy organic year-round is really tough. And they've come out with a list of the highest and lowest pesticide-containing foods. So the Clean 15 would be the list that you could buy non-organic that have the least amount of pesticide residues. Limit GMO foods. I just put that in there because honestly, we still don't know a lot about GMO foods. They may be the end all, for world hunger and all that, but
Isn't all our food genetically modified?
well, it's growingly, it's a lot, and most of it, doesn't have to be labeled, so we don't even know what we're consuming. Clean water. I use a Berkey myself. This is something, most municipalities, they only test for a select few number of, bacteria and other contaminants. They actually don't look at a lot of medications that get flushed down the toilet, or when you urinate or you poop, you actually poop out and urinate a little bit of that medication byproducts, and that actually doesn't get all filtered out. So we've seen studies that look at small levels of this, those medications in our municipality waters.
What's a Berkey?
It's a filter, a water filter. It's just a brand name.
Like Brita.
Yeah, but better. Regular exercise, as we saw in the study with the bike, increase overall activity. There's actually a study, I think it was in Australia a couple of years ago, where they showed if people sat for 6 or more hours per day, irregardless of their activity level, they had a 50% increased risk of all case mortality within the next 15 years. And that's like the big part of that was irregardless of their activity. So if they sat too long, that was their biggest risk factor for all-cause mortality. Okay, so sit less, be more active. You could sit for 6 hours and go run a marathon. It still didn't decrease your risk there. You still had the same increased risk. So be more active, sit less. Sitting is the new smoking. It's the new tolerated, vice, right? Clean air. Well, that's a big one right now. Go buy yourself a HEPA filter if you haven't already. And then proper supplementation. If your diet lacks in some of these methyl donors, then this may be a case where supplementation could benefit you. I'm not a big proponent of putting everybody on tons of supplements. I'm very minimalist in that regard. Take what you need to, and that typically doesn't mean take it all the time. Take it as you need it. Sometimes we're better at our diet than other times, and those are good times to supplement. Okay? Or if you have some of those genetic variations, I'm a compound heterozygous MTHFR, so I take methylfolate and methyl B12 almost every day.
What does that mean?
Those are
You are a what?
so I'm a compound heterozygous MTHFR, which just means that I have 1 out of 2 variations on 2 different significant genes, okay, which can affect my ability to make those methyl donors. Okay. So I want to give my body plenty of that support in the B12, folate, and all that. Okay. Testing-wise, there are a lot of companies out there. Oh gosh, if you just sit on Facebook for 5 minutes, you'll see 10 of these pop up. This is the one I've used for the last 8 years. It's great. I really like it. They actually have 30 MD, PhDs on staff that are always going through the literature continually looking at, and they'll rate each thing that they respond to, each gene that they're looking at. They'll actually rate it on a scale of 1 star to 5 star based on the amount of documentation and literature out there available for that particular gene. So this is you can see the stars on the right-hand side. So it'll list out the gene there and their scientific strength.
How much does it cost?
This one here is right about $400 for this. This is what's called a healthy weight.
How do you figure out somebody's healthy weight?
Well, this particular test is just looking at ways to improve your weight, lose weight, to modify your weight, healthy. So it'll focus on nutrients, possible nutrient deficiencies that you may be predisposed to, what particular diet that you may be better for, even exercise-wise, what you may respond to best. This is the report that I used in that example of when we first started about the lady who was a fitness instructor, who needed to exercise less and eat more. Well, we ran this test on her and it showed that she didn't respond well to cardiovascular exercise, like high intensity stuff. She would respond better to low intensity weight training. And that's what she did, and that's how she lost weight. So it does kind of help us direct some of those lifestyle modifications better.
The people that they perform these tests on, are these people with typical medical conditions, or are they being used for performance?
Yeah, I've got a lot of CrossFitters and other people that are just wanting to optimize their health. So trying to find that little edge, in whatever way they can. So yeah, we'll do testing on those too. These are some of the genes that we look at on this particular test. One's called eating disinhibition, and this is your increased tendency to eat more than normal as a response to a stimulus. So if you sit down in front of the TV and eat, you're going to shut off that stimulus to tell your stomach that you're full, okay? Or if you eat while you're driving, again, you're not going to get that signal that your stretch receptors in your stomach have been initiated and you're full. You're not going to get that. Food desire, this is a big one. Food desire is talking about how much are you willing to go to get that ice cream at 10 o'clock at night? Are you willing to drive 10 miles down the road to the convenience store? That's what food desire is about. Satiety, this FTO is actually called the fat gene. So this inhibits your ability to feel satiated. So you never feel like you're completely full. So you just continue to eat even though you've gotten enough calories in your diet. This leper one, snacking, this affects your leptin hormone, which has to do with your ability to regulate your food intake as well. 23andMe, I still use this a lot just because it's really cheap. It's $100 to $200 depending on which one you want. I do use software called Strategene. This extracts that information that we need that has to do with your methylation cycle, and then I interpret results based on this. And then there is testing now available to actually look at your methylation cycle and some components of that. So this is one I use, doctor's data. They will test your methionine, homocysteine, some variations there. So you can see, before I start just looking at genes, because we never just treat what's called a SNP, a single nucleotide polymorphism, which is that variation. You don't want to just treat that because there's so many other variables that we don't know about, we don't understand. We want to see if theoretically if this person might have a problem with their methylation. Now we can actually test and see if they're having problems with their methylation. And that's where this test comes in. And it gives us some ideas of which enzymes are not holding up to par. Some other useful links. MD Anderson is actually doing a ton of good stuff on cancer epigenetics. Actually, epigenetics, the first kind of application was for cancer. It was identified that colon cancer genes, or the tumors, actually had significant areas of hypomethylation, and that's where this epigenetic expression really started to kind of take off. That was back in the mid-'80s, I believe. Ben Lynch, this is where I've done some of my training. He actually graduated 2 years ahead of me in school. He's wrote a book called Dirty Genes. He's kind of one of the foremost experts on this particular MTHFR gene, which, if you remember, you go back to that methylation cycle, that was the key part of that cycle. University of California, they've got some really good TV lectures on this. This MTHFRsupport.com is great. SNPpedia is wonderful. So if you get your 23andMe results, you'll have, with each gene, you'll have what's called an RS number. That's its like ID tag. You can take that RS number and put it into SNPpedia, and it'll pull up every PubMed article that's ever been published on that particular gene. So it's a great resource if you really want to dive into some of this. Live-Wello, PBS, there's some other books. Nutrigenomics and Nutrigenetics and Functional Foods and Personalized Nutrition. That's an exceptional one. Okay. And then I love this quote by Dawson Church. The code imprinted in our DNA, the one thing we thought was for certain is just waiting for direction by us to change, creating a civilization that brings health, happiness, and vibrancy in ways in which the current medical establishment only dreams of. Health is your decision. Every decision you make throughout your day is directing the health of your body. And that's it. Okay. All right, I didn't get a lot of questions, so I know you're holding them, right? Yeah? Okay, yes, ma'am.
I just have a comment to make as I look at what's happening in our, with the separating of the families and holding children in our immigration centers and our detention centers, and we're looking at lack of good quality food, we're looking at sleepless nights, we're looking at high levels of stress, we're looking at so many of these things, like lack of exercise. We're looking at so many things that are affecting their health forever, probably.
Right. Yeah.
Unless those things
And their
get-
children and their children's
and their
children.
and their children. So this
Absolutely.
is really a crime.
Yeah, absolutely.
So the whole thing's a crime.
Okay, any other questions?
It was about that website, SNPD, was that .com?
Yes.
Okay, and then one other comment. Somebody said, aren't all foods genetically modified? No. And if you buy something that's organic, it's guaranteed by law to not be genetically modified. And so that's why a lot of people like me choose to buy organic, because we don't want to get something that's genetically engineered or genetically modified.
Right. Very true. Yes, sir.
Yeah, a couple of things. Can we get this online so we can,
Sure.
and how do we do that?
Yeah.
Go to your,
It will be available in about 2 weeks on the summer session.
Okay.
but that's a picture of it. I'm wondering if we could just download a PDF that,
Oh, I see what you're saying.
Yeah, let me give you my email. Just shoot me an email and I'll send it to you.
I'll give you the other thing is, if you, the SRC gyms, you work out, MRSA is epidemic. And now they've got posters all over the gym, because if you go to a hospital, you're gonna get MRSA, it's a good chance. If you go to the gym and work out, there's a good chance you're gonna get MRSA. So I understand meat's a bad idea. MRSA started with swine farms in the United States. However, so what do? You eat plants, okay? And yet, California frowns on Roundup, and yet 100% of the wines in California have glyphosate. 92% of Americans have glyphosate in their urine. How can we find out what we eat, what's in what we eat?
Well, that's where you grow as much as you can, kill as much as you can, and hook up with a local farmer if you need to. I know it's harder here, but honestly, that's why we started raising rabbits, chickens, goats, quail, all that sort of stuff myself, because I didn't really want to buy meat anywhere. I didn't know what it was exposed to, what it was fed, all of that. So try to go back to where your grandmother ate and what they did for food. That's going to be the safest thing you can do. If you can't do that, then you try, buy organic as much as you can, eat a plant-based diet as much as you can, and then any animal products that you do eat, you want it to be highest quality.
The plant-based diet doesn't really work because the plants are, like the meat, are polluted. So one suggestion maybe is you take home stuff from the store and you take a picture of the barcode. And it used to be illegal to go to a website to explain to you what was in it. Now they exist. So stack up the barcodes on your smartphone or whatever, and go to the website that tells you what's in it, and other websites that say they're lying, this is what's really in it. Do you know a better way to do this?
Buy organic.
My grandma
yeah.
could not eat what my grandma ate right now.
Oh yeah, no, I totally get it.
That's not, yeah.
Yeah, there's an anthropologist out of, I think it's UC Davis, that estimated that about 72% of the food that we eat now, like our genes have never seen in human history. So our bodies are still trying to figure out what the heck to do with that, because it's something, there are proteins and things we've never seen in existence before.
You can't escape it. For decades, I spent 5 months a year out of the country. I eat all my meals in restaurants out of the country. I eat all my meals home here, and I try to eat well. My body does not like being in Alaska or America. The same thing, if people come here, foreign exchange trip, you're gonna gain 5 kilos and come down.
Yeah.
So it's food in the United States is, forget it.
Yeah.
You can't get away from it. So what's the solution to,
As you said, I think you can grow as much as you can.
And put pressure on our elected officials to
Absolutely.
make our food supply cleaner
Yeah.
and safer, because we have the most poisonous food supply in the world.
You can't get your elected officials in the same city right now.
That's true, but I mean, they are the ones who have to respond to us, so if we are not happy with having the most poisonous food supply in the world, and I'm not, then we have to put pressure on them. This is important.
Absolutely.
I don't want to have to spend every dime I earn to buy organic food, but health is where I spend my money.
Yeah.
And so it's very frustrating. It's really frustrating. And where do those toxic chemicals go in our body? Our bodies can't handle them, so they're stored in fat.
Yep.
You know, it's to protect us.
Yeah.
And we don't want the fat.
Yeah. Yeah.
It's just this circle.
And that's just it too. Like you want to be able to support your body's ability to eliminate those particular environmental toxins too. So staying as healthy as possible, providing your body the nutrients it needs for proper, liver detox, sweating a lot, using your skin to detox, all those kind of things help. Yes, sir.
I wonder if there's hesitation to make remarks about epigenetics, being that there must be so much variation, even among just the group of people that we have in this room. When you shared the story of somebody that, to me that's like violating the second law of thermodynamics. You bring in more calories, you exercise more, and it doesn't help. I mean, to me that's a shocking story. And granted, it might be an anomaly, but there has to be so much variation in a population of people that to make a statement that this is good, bad, or indifferent is kind of hard to make.
Sure.
When you're talking to an individual who hasn't had this stuff mapped out.
Yeah. Well, the variation is actually less than we think. It's 0.1% difference between one person and the next. It's like typically one base pair in 1,000 that you're going to see a significant difference there. So it's less than you think, but yeah, you're right. That's where testing needs to be available. And I think you'll be able to go in, get tested to see what genes are being expressed, start a particular lifestyle modification type of program, and then retest it in a month and see what type of genes are being expressed at that point. Like, I think that's in the future, in the near future. 5-10 years, I think that'll be available for us to be able to do. And then you'll be able to see, that's personalized medicine for you. Be able to see what nutrients you need, what medication you can and won't respond to. Like, I think that's the future of medicine right there. Yes, sir.
If someone were to take the medical supplements, how would you ensure that they're silencing the right things?
You're not. Yeah.
So you're just hoping?
Exactly. Yeah, you're hoping for the best there. Now, for the most part, we know, and this is the tricky thing with cancer too, at the beginning, we saw this, like in colorectal cancer, we saw a significant amount of hypomethylation in most of the tumor cells that were looked at. But then we started seeing these hypermethylated segments in a few of the cells that had some significance to them as far as the type of tumors and genes that they were, or proteins they were expressing. So this is still a part that we just don't know enough about. And that's where I'm not a proponent of taking 20 milligrams of methylfolate a day. Like, that's a huge amount. Typically I'm doing 1 to 2 maybe milligrams of that per day, if that. And then I check their methylation status, like that doctor's data test I was showing you earlier, just to make sure like things are moving properly. We don't want to speed things up too much, and we don't want to slow things down too much. So we're trying to find that sweet spot. And honestly, we don't have enough information right now to know exactly where that sweet spot is. But if we try to fall somewhere in the middle of the range, even lower the range, we're likely to support that process without over, supporting it, hypermethylating it. Does that make sense? Yeah. Sir.
Yeah, I got a decent pollution device, and it measures PM 2.5, and at my place it's usually between 5 and 10. So in the last couple of weeks, it's routinely over 100, up to 500.
Wow.
Have you been able, and this is what we're breathing, and I definitely feel it.
Yeah.
Have you been able to test the effects of this smoke and pollution on our epigenetics?
No.
Is there a way to do that?
I'm sure there is. Maybe the university has the ability to, but it's not available direct to consumer yet.
Do you recommend places to test our epigenetics? Is this affordable?
Well, the only thing that we can use to really test our epigenetic expression is to look at that methylation cycle right now. That's really the only way we can kind of speculate as to how you're, and that's only one part of that. So that's just not available to us right now as a consumer.
What it is, would you test this stuff and let us know? how
Yeah.
it works?
Oh, I'd love to give them access to this kind of stuff, this technology, and be able to just experiment with it. You know, yeah. There's a question. Yes.
Hypothyroidism, which was mentioned at the beginning, has been growing, especially for women. And yet, in the 50s, when they added iodine salt, that was going to be the answer for it all. What are your thoughts on that and why?
Well, I've seen the thyroid is the most sensitive gland to your environment. Like I said earlier, our environment's just growing increasingly toxic. So it's no wonder the incidence of thyroid conditions is going up. Same with thyroid cancer. Those incidents are going up as well. I mean, I see patients in their 20s and 30s now with hypothyroid, like just frank primary hypothyroid. Their thyroid's just giving out, not working, and you didn't really used to see that.
And so the answer is Synthroid and Cytodal and...
Well, I think that's a symptom a lot of times of what's going on deeper. Is that a response of your body to your environment? And your body is trying to downregulate metabolism because it feels stressed. And that's usually how it's worked in the past. I measure something called reverse T3. So, T4 is what your thyroid normally makes about 90% of. T3 but is the active thyroid hormone, right? And T4 gets converted into T3 mostly in your peripheral tissue, mostly in your liver, about 60% of it, in your gut and in your muscles. But your body in times of stress will convert that T4 into something called reverse T3, which looks very similar to T3, but it just doesn't do anything metabolically for you. And your body will do that in a response to stress because it's trying to downregulate your metabolic rate. It doesn't know if you're going through a famine, you're running from the bear, or whatever stress it is. Your body sees that stress as the same thing. So it's going to downregulate your metabolic rate in response to that to conserve energy. And I think for most of us, we're a society that eats too much fast food. We don't sleep enough. We don't move and exercise enough. And we're just stressed to the max all the time. You couldn't design a better experiment to make someone unhealthy. Okay? So our body is stressed in multiple avenues there. And I think our bodies are trying to respond to that. We're trying to downregulate our metabolic rate and other things to try to help us survive longer. Because if we did that and we're going through a famine, if we downregulated our metabolic rate, we survived. If we didn't survive. So your body's learned from your ancestors of how to respond to stress, and that's what it's doing. Yeah.
How specific are those stressors to specific stimulus? So we talked about there's cardio. I mean, you could be resistance training, sports. I mean, are these stressors specific to the activities, or do they just seem as stress and stress period?
Well, I think that has to do with your genetics as well. But if we had to kind of draw a blanket statement, I'd say intense cardiovascular exercise for greater than 30 minutes typically creates a stress response. But exercise in general, you won't elicit a bigger free radical response than anything else besides exercise. But your body responds to that by producing more antioxidants and other things to help quench that. So there's a balance like everything else. I typically tell people if they come in and we see labs that show that their body's stressed, so the cortisol levels are high, the reverse T3 is high, obviously their body's stressed and they're having a hard time losing weight or whatever else, sleeping, whatever else it is, then I tell them, limit the cardio. Do something really light to moderate, lift weights more, do cardio less in this time period until your body realizes it's not under an immense amount of stress. That's the hard part to get people to do. This is pretty typical. I see plenty of business owners, CEOs, high-power type A guys who they're working 80, 90 hours a week and then going on the weekends and they're riding their bike 100 miles. They're just taxing their body and they're wondering why they can't sleep at night and they can't lose weight and they're getting a belly on them and all that kind of stuff. It's just their body's stressed and it's responding to that. Yeah. Yes, ma'am.
If 10 hours of sleep is the optimum, how much is too much?
Well, I wouldn't say 10 hours is the optimal. There's actually some evidence that anything above 9 could be counterproductive in some people. So somewhere between 7 and 9, I think, is sort of the optimal range. But again, that's different from one person to the next too. I've listened to a lot of sleep specialists, and maybe Clay will talk about this next week too. But if you want to find kind of what your optimal time to sleep is and amount, go on vacation and just go to sleep when you feel tired, wake up when you feel like you want to, and then that's kind of your time period that you need to try to adjust your schedule to. Yeah. Yes, sir.
Do you have any thoughts on maintaining muscle mass as you age?
Yeah. I think the one thing you could do to improve your longevity and quality of life is to have as much muscle as you possibly can going into your later years. That's going to be tough because a lot of that's hormonally driven. And, as men and women, we lose a lot of our sex hormones, after the age of 35, typically those are starting to go down. So you want to try to, lift weights as much as you can. Nutritionally, actually you need more protein as you get older too. So you want to be getting somewhere between like 0.38, 0.42 grams of protein per pound of body weight, somewhere in that range. But you need the stimulus too. You need to be pushing some heavy weight. Yes, sir.
How about naps?
Yeah, that's tricky based on who you talk to. What about naps? Are naps good, bad? They're good. All right, we've got a consensus here. Moving on. I think for someone who's having some circadian rhythm issues, naps can be detrimental, especially a nap longer than 30 minutes. You can start to get some hormonal changes there with melatonin and other things. So that can be tough. But plenty of other studies have showed napping for less than 30 minutes can be stimulating and effective. So I think like anything else, there's a balance there you have to figure out. I can't nap. If I nap, I'm done. I'm done for the day. But other people, they feel rejuvenated by it. So again, I think it's individual.
So I'm a school teacher, and I think I've shown like that PBS video on epigenetics, and it wasn't even in my vocabulary until maybe 5-6 years ago.
Yeah.
Are there other fields? Has epigenetics contributed to any other fields such as evolutionary biology or anything, or is it specific to health, or at least what we're looking at right now?
That's a good question. I honestly don't know. Most of the stuff I see has to do with medicine, personalizing medicine, cancer therapies, things like that. So I don't know if it's being applied in other avenues or not. I'm sure it probably is, but I'm just not aware of it.
I could think of psychology and trauma.
Psychology and trauma? Oh yeah, I could definitely see where it could be useful there. Absolutely.
You're hearing about that more in
Yeah.
trauma response and
Right.
rehab.
Yeah. Any other questions? Yes, sir.
So if you have an exercise routine that that elevates your heart rate for an hour a day, that's excessive.
I wouldn't say it's excessive. It depends on how high you're elevating it and for how long you're elevating it. What percentage of your max heart rate are you getting? And what your hormonal response to that is too. Sometimes, some people, if they're well conditioned, they can be at 70-80% of their max heart rate and they're not eliciting a stress response. Whereas other people, they get above 120 and their cortisol levels are already starting to rise. Now, you wouldn't know that unless you tested yourself around that, but you just kind of assess yourself and how you feel. If you feel drained after exercising like that, where you need a nap, that was probably too much. Or if you feel the effects of that for another day or two, then it may have been too much for you. You want to try to find that sweet spot of where you do exercise and it's stimulating for you and you feel better for it the next day or two.
Tired for 3 or 4 hours a little bit, but then the next day you still
Yeah.
it's okay to go again.
Right. And you look at, risk factors too. Like, I've seen people who, go, they do a lot of cardiovascular exercise, and then we look at their ultrasound of their heart blood vessels or their carotid blood vessels, and they're laying down a lot of plaque. They've got a lot of inflammation there. And it's like, obviously that level of exercise is actually becoming detrimental to them. It's creating more inflammation than their body can actually handle. So. Any other questions? Yes, ma'am.
I keep hearing a lot about stress. I wonder if someday maybe stress will be the new smoking.
Yeah, I think it already is. Oh, yeah.
Are you doing any testing on that, or is there anything on the stress?
I wish testing, there were better available tests for the response of stress. Right now I look at some adrenal hormones and like some of the reverse T3s and things like that. Look at sex hormone suppression. So if someone's, I see this quite a bit actually, 20, 30-year-old, fatigue, they come in, we measure their sex hormones and they're actually really low. Like your sex hormones shouldn't be that low at that age. So obviously that's a sign of chronic stress there. Again, their body, if your body thinks it's running from the bear, making progesterone as a woman, which is that progestational, pro-pregnancy hormone, goes down on the list. For a man, testosterone goes down on the list of priorities, right? It's making stress hormones like adrenaline and cortisol to get away from the bear. And that doesn't correspond with procreation. So you can have one or the other. You can't have both at the same time. You don't want to procreate and run from the bear at the same time. That's just not a good idea. Yes, sir.
You mentioned the 6 hours of sitting.
Yeah.
So what if, are the studies so specific that it's complete sitting, or if you get up for a couple of times, walk around the office, come back down, is that, is it fine-tuned enough to know whether that has an effect?
Well, in the study that I referenced, they didn't say much to that, like how much these people got up throughout the day. They just said 6 hours or more of complete sitting. So I'm assuming they measured the complete time that they were actually in a seat. They did speculate, though, that people who stood, even if they were stationary, didn't have the same risk. But, yeah, I don't know. Yeah. My point is just get up and move as much as you can. Yeah, absolutely. Stand as much, sit. If you're going to sit, get up and take breaks as often as you can. Don't sit there for 3 or 4 hours at a time. Because, one, it's not a good position for your body anyway. Like, our bodies were not designed to sit. It's just, back pain, neck pain, all of that. It's just not a good idea. So, for a number of reasons.
Cut it off at one or two more questions.
Okay. Yes, sir.
Okay, so in terms of overtraining, because I can't measure my cortisol levels every time I work out.
Sure.
So I played football through college my whole life, and now I've moved into ultra running and stuff like that. My only indicators are performance.
Yeah.
My time was slower, or time was faster, and what do I attribute that to?
Yeah.
Or am I sore today, or am I sore for 2 days after that last training session? What are some signals that we can know, that I can know myself without going through a doctor to find out if I'm overtraining?
Yeah. So there's some great wearables that are available now to kind of test some of that out, like heart rate variability. Heart rate variability has been used to assess like recovery for athletes for a while now, but most wearables are starting to calculate that for you. And what heart rate variability is, say your heart beats 60 times per minute, right? You would think each heartbeat is about 1 second apart, but that's not true. There's some variation between that one heartbeat and the next heartbeat. If your heartbeat is double that, 120 heartbeats per minute, now you're going to have less variability because you have less time between heartbeats, right? So typically, the lower heart rate you have, the more variability you have. And so there's a way that you can test this heart rate variability to see what your recovery is and how your autonomic nervous system is functioning. So your autonomic nervous system, you have two parts of that. You've got your sympathetic, the fight or flight, and your parasympathetic, your rest and digest part, okay? And those also affect your heart rate, blood pressure, and all those automatic processes in the body. So heart rate variability looks at heart rate and how much that's varying and determines how well you're recovering. So that's one I have a lot of my athletes check, and it helps them. I've been checking it myself for the last 3 years, and I can tell you, if I wake up in the morning and my heart rate variability is low because when I'm stressed, I didn't have a good night's sleep, I'm not going to go to the gym and push myself really hard that day because I know like my recovery is probably going to be pretty poor. But if I wake up and it's really high that day, then I'm thinking, okay, this is a day I can actually push myself and get good results from it too. So that's an easy one, pretty cheap.
What's your wearable that you know your heart rate variability?
I use my Apple Watch,
Oh.
and there's an app on that I use for it. Yeah.
Do you use the Oura Ring also?
No, but Jeff, he's my health coach, and he uses it. He's seen some good stuff with it. Yeah, I think it also measures body temperature too, and your sleep quality. So there's some other ideas, like if you had poor sleep, you know, maybe a night or two after you had a hard workout, maybe that could be an indicator too. So you just, what's that? The Oura Ring. Oura Ring, yeah, it's a ring that you can wear. It measures heart rate variability, your sleep pattern, temperature, some other things I think too.
If I want to find out more about this, should I research heart variability?
Heart rate variability, yep, yeah. There's some free apps like HRV for training, Elite HRV, Heart Math is probably the leader right now, and they've been around for a long time. Yeah. Okay. Garmin, yep, Garmin's wearing they're making a few wearables now that track HRV.
I don't want to shoo anybody off, because there seems like there's still a good conversation going, but I'd like to formally thank Dr. May
Jeff,
for
thank
that. I'm sure he'd be willing to stay for a bit.
Yeah, sure. If you have questions, come on down.